Hi students, this is Professor Lisa Schimmel at Crafton Hills College, and today I want to talk to you about specific defenses of the immune system. Let's start by doing a little overview of the two main components of the immune system. First is the humoral immune system, and that involves the actions of a type of white blood cell known as a B cell. Now, when B cells are exposed to antigen, and I'll define antigen for you in just a few minutes, but anyways, when B cells are exposed to antigen, they undergo some further development and become what are known as plasma cells. These plasma cells are the ones that actually produce antibody molecules and secrete them into the environment. The second component of the immune system is called the cell-mediated immune system. This involves a type of white blood cell known as T cells, and there are 4 different types of T cells, and we'll talk about what they are and their functions a little bit later on in this lecture. All right, what I'd like to talk about now is what's called acquired immunity. And as the name suggests, this is immunity that we didn't always have. We picked it up, we acquired it at some point in our lifetime. And there are 4 categories. The first of these categories is called naturally acquired active immunity. We refer to it as naturally acquired because this is something that occurs in our day-to-day life. Every day of our life we are being exposed to a variety of antigens in the air that we breathe, food that we consume, contact that we have with other individuals and surfaces. And we refer to it as active immunity because in response to those exposures, our body does actually a number of things, but 2 main things. One is our body will produce antibody molecules that are specifically designed to attach to and help us eliminate or destroy those antigens. The other main thing our body does is by producing what are called sensitized T cells. And again, those are T cells that are going to be designed specifically to interact with that antigen and help to eliminate it either through phagocytosis or the production of toxins like cytokines, for example. Why don't I take a moment here, even though it's a little bit, this is going to be found a little bit further on in your outline, let me go ahead and define antigen for you before I move on. Antigen is anything that's foreign to the body, and that could be a microbe like a bacterium or a virus. It could be dust or pollen, mold, dirt, animal dander, anything that's foreign to the body and that also elicits an immune response, production of antibody molecules and sensitized T cells. Okay, now we're still talking about naturally acquired active immunity. How long does this immunity last? Well, there really isn't a simple answer to that question. One factor is that the effectiveness and the duration of this immunity is going to vary from one person to another. And it's also going to vary depending on the particular antigen or pathogen that we happen to be responding to. Let me give you some examples. Immunity to measles and a viral disease called yellow fever and also chickenpox is typically considered to last an entire lifetime. So once you've had one of those diseases, hopefully if you've been vaccinated, you should be immune for the rest of your life. Now, in some cases, the immunity that we develop in this manner will be short-lived but not lifelong. There's a bacterial respiratory disease called diphtheria. We'll talk about it in an upcoming chapter. If you become infected with this organism and develop the disease diphtheria, upon recovery, you'll have what we call partial immunity. That means that you could become infected again, but any subsequent infection should be somewhat milder. It'd be a whole lot better just to get vaccinated, I think. And then in some cases, we really don't effectively develop any lasting immunity, or sometimes we say durable immunity. For example, there's no immunity to gonorrhea, nor is there really any effective immunity against colds. We talked about that when we discussed the viruses. And a form of pneumonia, bacterial pneumonia called pneumococcal pneumonia, we don't really consider people to be immune to it upon recovery. They can't get that infection again. All right, let's move on to naturally acquired passive immunity. Let me give you a definition and then we'll talk some examples. All right, this occurs upon a natural transfer of antibodies from an immunized donor to a non-immunized recipient. I've got two examples I want to discuss. The first one is called placental transfer. Here's how it works. Pregnant woman, anything that she is immune to, either through natural exposures on her own, or perhaps she's been vaccinated against them. Some of mom's antibodies will cross the placental barrier and be received by the fetus. So we call it passive immunity because the fetus is not producing the antibody molecules, mom is, all right? And those antibodies, after the child is born, will provide the newborn with temporary immunity. Usually it lasts about 2 months, about 8 weeks. And that just gives the child's immune system a chance to kind of fire up and start to respond to its own exposure. So it's just a good means of temporary immunity to provide some short-lived protection for the newborn. The other example of naturally acquired passive immunity occurs through the ingestion of colostrum. And colostrum is the name that we give the first breast milk. It's produced for maybe 24 hours after birth. It isn't technically milk. It's got a very high fat and sugar content and very high protein content, the majority of those proteins being antibody molecules. Now, when the newborn suckles, it's important and ingests the colostrum. It's quite important that the infant ingest that colostrum as soon as possible after birth. Here's why. Immediately after birth and lasting for, possibly 12 to 24 hours, the infant's GI tract, it hasn't really started functioning yet. For example, the digestive juices aren't really working quite yet. And the lining of the intestine is quite porous or permeable. So here's what happened. If the infant ingests the colostrum within this rather short window of time, 12 to 24 hours, the colostrum will pass through the stomach undamaged, enter the lower digestive system, the intestinal tract, where it will be absorbed directly into the bloodstream. Now, each hour that passes after birth, we're going to see a decrease in that porous nature of the intestinal lining. And after a critical point has been reached, let's just say 24 hours for our discussion, the absorption of the antibodies into the bloodstream will no longer occur. And even if it did, the stomach acids will be working now and the antibodies would be degraded and made ineffective as they pass through the stomach. So we need to get colostrum in the infant as soon as possible. All right, now again, to reiterate, that immunity is short-lived, a couple months at most. And while we're still on the topic of naturally acquired passive immunity, I wanted to mention a concept known as vaccine interference. Here's what I mean. If we vaccinate too soon, the antibodies that the infant received from mom through placental transfer and ingestion of colostrum, they will interact with the vaccine and render the vaccine ineffective. I don't mean that this is going to have a harmful effect on the infant other than the vaccine not doing its job. But so we've got this very critical period of time in the life of this newborn. At about 8 weeks of age, the passive immunity that the child has acquired from mom is beginning to decline. Certainly their immune system has at least begun to function at this time. The child is producing its own antibody molecules and sensitized T cells. But like I said, this is about the point where we're going to start doing most of our vaccinations. I do acknowledge there are some, for example, hepatitis vaccine that may be given at birth. But for the most part, we need to wait until about 8 weeks of age to begin vaccinating our infant. Okay, let's move on to the third category of acquired immunity. This one is called artificially acquired active immunity. All right, now this is the type of immunity that you develop when you're vaccinated. So we call it artificially acquired because you didn't just naturally become exposed to the antigen. It was injected into you, or perhaps you inhaled the vaccine, as is true of some flu vaccines, and there are also even oral vaccines. So anyways, the antigen was artificially introduced into your body by one of these means. But the immunity that you develop is indeed active because in response to the vaccine, your body does all the things that it would do upon a natural exposure. It produces antibody molecules specific for that antigen, and also the sensitized T cells that we talked about. Now, the fourth category of acquired immunity is called artificially acquired passive immunity. Okay, here's the definition. This type of immunity occurs upon injection of antibody molecules obtained from an outside source. Let me explain with an example. Let's say we have a patient that has been bitten by a venomous snake. The snake's venom is a very fast-acting neurotoxin. And it isn't that the individual's immune system wouldn't respond. It most certainly would produce antibody molecules, sensitized T cells, but the venom is much faster than the immune response. The immune response takes days to weeks to come to completion. And so this individual would die from the effects of the venom long before their immune system could do anything to protect them. So we use what's called snakebite antivenom to treat people that have been exposed to snake venom. Here's how it's made. It's produced usually in a large animal. Often a horse is used. And the same horse may be used for years or decades as a source of snakebite antivenom. The horse will be periodically injected with snake venom. Now, we certainly aren't trying to kill the horse, but rather to stimulate its body to produce antibody molecules against the venom. And then also periodically we will harvest blood from the horse. Typically they take out about a gallon of blood at a time, which is kind of the horse equivalent of a human pint of blood. Now, the horse's blood will be centrifuged. That means spun at a very rapid rate in tubes or other containers. And the heaviest cells, the red blood cells, they'll be found at the bottom of the tube. And then above those we'll have white blood cells and platelets. And then there will be a kind of a pale yellow clear fluid called plasma or serum, and that's where we're going to find the majority of the antibody molecules in that, let's just call it serum for this discussion. That's the part that will be removed and is literally the snakebite antivenom. That's what will be injected into the person that was bitten by the snake. Now, the only potential problem here is if our patient is also allergic to horses, in which case they could experience an anaphylactic reaction in response to the horse serum. And anaphylaxis refers to a very severe, potentially life-threatening allergic reaction. So anyways, I guess if you are allergic to horses, you shouldn't get bitten by a venomous snake. All right, let's go ahead and move on in our outline and talk a little bit more about serum. You've got a diagram in your notes that shows a test tube with some blood in it, and that blood's been centrifuged. So you can see, as I described a few minutes ago, the red blood cells on the bottom, platelets, white blood cells, and then the serum. Now, like I said, most of the antibodies in blood are going to be found, excuse me, dissolved in that serum. Now, serum is actually a very complex substance. It's got numerous components, and there is a technique that we can use to separate the components of serum. It's a process called electrophoresis. Now, electrophoresis involves taking our sample, placing it into a small well that's cut into an agarose gel slab, and then we are going to apply an electrical current that's going to be used to push our sample through the agarose gel, like from left to right in the gel. And the various components of, in this case, serum, will separate out based on their size and their electrical charge. When we do this, we would discover that serum is made of these main components. One portion, or sometimes we refer to it as a fraction, is called the alpha fraction. We also have a beta fraction, a gamma fraction, and that's the part of serum where we find almost all of the antibody molecules. And then finally, another component called albumin. All right, so those are the components of serum. Now, I think I should mention at this point that the terms antibody and immunoglobulin are used synonymously. AB is used to abbreviate antibody, and IG is the acronym for immunoglobulin. All right, I think I've already defined antigen, any substance that's foreign to the body and that stimulates an immune response or elicits an immune response. And let's move ahead and take a look at our typical antibody molecule. So you've got a diagram showing you 4 main parts of this molecule. We've got 2 longer sections. They're in the center of the molecule. They're referred to as the heavy chains because they're larger. And then one on each side of the heavy chains, shorter chains called the light chains. All 4 of these chains, the 2 heavy and the 2 light chains, are bound together by means of disulfide bonds. Now, there is a flexible region called the hinge region, and that allows some movement in the molecule to accommodate different sized antigens. Now, if you take a look at the ends of all 4 chains, we have an area there labeled as the antigen binding region. All right, this is a little silly, but think of my fingers as the antigen binding regions. Those are the parts of the antibody molecule that are actually going to come into contact with our antigen and bind to it. And so if it's a large antigen, we may need to spread the antigen binding regions further out. If it's a smaller antigen, then that hinge area is going to move so that the antigen binding regions can grab a hold of that smaller antigen molecule. The parts labeled variable region, and that would be labeled A and B on your diagram, those are the parts of the molecule that will be designed to attach to a specific antigen. So for example, you have some IgG molecules that were designed to interact with that cold that you had a month ago. And you've got other IgG molecules that can interact with the salmonella that you ate in some undercooked eggs. And one IgG molecule can only attach to the cold virus, and the other can only attach to the salmonella cells. So they are actually very specific. Now, the portions labeled C and D on that typical antibody molecule, they are referred to as the constant region. And that part of the antibody molecule is going to determine what category or class of antibody molecule we have in front of us. And in just a moment, I'm going to talk about the 5 classes or categories of antibody molecules. One of them is called immunoglobulin class G. What I mean in reference to the constant regions is that all immunoglobulin G molecules would have identical constant regions. That's what makes them IgG molecules, and so forth. Okay, let's go ahead now and talk about the 5 categories of immunoglobulins or antibody molecules. And we're going to start with immunoglobulin class G, or you could say just IgG. All right, as it says in your notes, that class of antibodies makes up somewhere between 80 to 85% of the antibodies in serum. And it is what we refer to as being a monomer unit antibody. That means single unit. Looks identical to the diagram that we just discussed. And it's a relatively small molecule. It's the smallest of the 5 categories of antibody molecules. And this size allows it to do some really important things. One is that IgG molecules are small enough that they can actually cross the walls of blood vessels and leave the bloodstream or the lymph system and enter tissue fluids. That's important because not all antigens that we encounter are going to be found in the bloodstream. Some may be found in other parts of the body. So immunoglobulin class G can actually exit the bloodstream, move through tissue fluids, and arrive at the site of infection to interact with the antigen. Where was I? IgG. So, as I was saying, it can exit the walls of blood vessels, enter tissue fluids. Another important thing that IgG can do is cross the placental barrier. So this is the class of antibody that's going to be involved in that phenomenon known as placental transfer that we talked about earlier. Now, what does IgG do for us? Well, it helps protect against bacteria and viruses that enter the circulatory system. It also helps to neutralize bacterial toxins, and it enhances phagocytic cells. Now, what I mean by enhances them is, it makes them more aggressive, more highly phagocytic. And also, IgG is involved in some complement reactions, and you will have read about that in your reading assignment on nonspecific immunity. Okay, let's move on to immunoglobulin class M. Now that category of antibodies makes up somewhere between 5 and 10% of the antibodies in serum. And it is a large molecule. The M in this acronym actually stands for macro. It's a pentamer unit antibody. What that means is this. We have 5 monomer unit structures that are joined together by means of a mechanism called a J chain. So that we've got all of our antigen binding regions are on the outside of this complex, and IgM can actually bind to multiple antigens, and they form large clumps. That makes it easier for the other cells of the immune system to kind of come back through and clean up the mess. All right, now because IgG molecules are large, they are not able to cross the walls of blood vessels, nor can they cross the placental barrier. We know that IgM also enhances phagocytic cells, and it's involved in some complement reactions. Beyond that, we're not certain as to what other functions of IgM might be. All right, I'm moving on to IgA. Now, there are actually 2 forms of IgA. There is a monomer form, and that's the form of IgA that we find in blood and lymph. And it makes up approximately 10 to 15% of the antibodies in serum. It is less effective than the dimer form, right? Now, both forms of IgA, what they do is they actually coat the surface of antigen and help prevent them from attaching to our tissues. Maybe you'll remember earlier this semester I talked about bacteria that have capsules or pili being able to more effectively attach to us and proliferate to the point where they cause a disease state. IgA helps prevent that attachment. The dimer form of IgA, we've got 2 monomer units joined together, again with a J chain, and that dimer form of IgA is found in mucus secretions, such as mucus, tears, saliva, and breast milk. They also help prevent antigen from attaching, and this dimer form of IgA is in colostrum, and that's what the newborn infant is going to ingest to help booster their passive immunity, their temporary passive immunity. Again, so to reiterate, the main function of IgA is to prevent attachment of pathogens so that other body mechanisms, like in the eye, tears can wash them away, or they can be washed away by mucus or other mechanisms that we have. Okay, let's talk next about immunoglobulin class D. This particular antibody makes up approximately 0.2% of the antibodies in serum, and there's a whole lot about this one that we don't know yet. We don't completely understand its function. Let me tell you a little bit about it though. It is a monomer unit antibody, but it's bigger than IgG, so it can't cross the walls of blood vessels, nor can it cross the placental barrier. It is found, however, in blood, in lymph fluid, and also coating the surface of B cells. And I already mentioned can't cross the placental barrier and is not involved in complement reactions. And then our last category of antibody molecules would be immunoglobulin class E. This one makes up approximately 2/1000ths of 1% of the antibodies in serum. And IgE binds to white blood cells known as mast cells, that's M-A-S-T, and basophils. Those cells are involved in allergic reactions, and when IgE binds to them, it stimulates those cells to release histamines, and histamines are involved in allergic reactions. We know that we have had a lot of histamines released in our body when we have itchy, scratchy, runny eyes and runny nose, or perhaps a skin rash in response to something our body decided it didn't like. And people that have a lot of allergies, they will have higher concentrations of IgG than people that don't have a lot of allergies. All right, you guys, I'm going to break here, and we will pick this up with a discussion of T cells in just a little bit. Thanks for watching.